also doing some last checks before your PSPM. What else do I need to tell you? Oh yeah, itu lebih kurang begitu saja lah. Okay, so before I go on, how do you feel about your pra-PSPM? Was it what you expected kah? Lebih susah kah? Bikin kamu rasa macam tidak harapan sudah kah? How was it? How was the pra-PSPM?
Boleh lah, Miss. Sedang-sedang.
Boleh sedang-sedang. Hard but manageable. I think if I look back at the scores, mari saya tengok balik your scores. Based on your scores, where are your scores? Complete KOB. Ramai juga lah yang boleh disebabkan markah PB, but some of you score less than 10 marks on the whole thing. Macam paling tinggi saya nampak 27 kah out of 30 questions. So that's okay, but like thankfully your PB marks are quite high, but that doesn't mean you should relax too much lah. Try to do your best, lagipun macam ini saja peluang kamu untuk ada PSPM objective. So enjoy this chance of answering and tembaking if you don't understand. Okay. Let us then continue with sharing screen with the discussion for the pra-PSPM questions. Where is it? I need to set this up. I share this. Okay, so now I'm sharing this screen. Okay, cukup jelas. I'll try to read the text as I go through the question lah. So kalau kamu punya phone punya gambar tidak jelas pun tidak apa. We'll try to apa tu? Try to go through it. Okay. So saya akan tengok setiap soalan. Kalau soalan tu macam senang, I just move on. Right, so let's get started. This is the same questions that you had for your test last week, just that it is now in word form. Right, let's go right. Banyaknya saya punya bunga-bunga. Mari, start. Nombor 1. So question number 1. A carbon compound extracted from living tissues was found to contain exactly twice as many hydrogen atoms as oxygen atoms. To which of the following classes of biological molecules is it likely to belong? So untuk soalan ini of course relating to chapter 1, molecules of life. So you're starting to identify a carbon compound. So that means this molecule contains some carbon atom, contain twice as many hydrogen as oxygen atoms. Maksudnya dalam molekul ni ada karbon, ada hidrogen, ada oksigen. Bilangan hidrogen lebih banyak daripada oksigen. So it's gonna be CHO, twice as many, 2 kali ganda lebih banyak. So this is the empirical formula for carbohydrates. And if you know, glucose punya molecular formula is C6H12O6. Okay, so the class that this biological molecule belongs to is carbohydrate. If you have any questions, you can type or ask using voice right away. If you don't ask anything, I'm going to continue with the next question, all right? Supaya kamu tidak di sini lampau lama. Okay, so that's it for question number 1. Now I move on to question number 2. Question number 2, the research of Erwin Chargaff, I don't know how to pronounce his name, was essential in the discovery of the structure of DNA by Crick and Watson. Chargaff analyzed the base composition of DNA from a wide range of organisms. He reported his findings using the initial letter of each base to stand for the number of that base found in organism's genome. Which of these relationships did he find within the genome of each organism he studied? Rumusan dia adalah, dia buat eksperimen, mana satu daripada statement ABCD ini yang benar mengenai eksperimen dia. So, eksperimen dia ni dia kaji, dia dapat tengok bilangan setiap base dalam DNA. Before we go into the answers and the options, it's okay if you don't know who the heck this guy is. You don't have to know him to answer this question. You only have to remember what you know about DNA. So, saya tahu kalau DNA ada adenine, guanine, cytosine, thymine. Dan saya tahu setiap daripada mereka ada pasangan dia. So, adenine will pair with thymine. And cytosine will pair with guanine. So I know in any DNA that I look at, the number of adenine molecule in that DNA is gonna be the same as the number of thymine molecules in that DNA. Contohnya, kalau DNA saya adalah 20% adenine, dia akan mengandungi 20% thymine. Cytosine and guanine will also have the same number of molecules. So if my molecule of DNA has 30% cytosine, it will contain 30% guanine. So A will be same as T, C will be same as G. A will not be the same as C. A will not be the same as G. T will not be the same as C. T will not be the same as G. So, now we look at each of these options. Tengok mana yang dalam pilihan ni paling ngam lah, paling sesuai dengan apa yang kita tahu pasal DNA. So, this one confirm tidak boleh. This one confirm tidak boleh. A and D are of course out of the question. The only choice is between B and C. So in this one, the answer is going to be 2C. Any problems with question number 2? Okay, I'm moving on to question number 3. So in question number 3, we are looking at, this one is already going into chapter 2. 3 plant cells X, Y, and Z are in contact with each other. The water potentials are shown in the table. Which option correctly describes how water will move by osmosis between the cells? So if I get question, what I like to do is I start drawing. Sudah saya paksa kamu melukis kan? Saya memanglah tahu saya suka melukis. Okay, so I'm gonna draw 3 plant cells next to each other. This is plant cell X. This is plant cell Y. And this one is plant cell Z. Okay. I'll focus on this one a bit more. Cell X will have water potential of negative 500 kPa. Cell Y will have water potential of negative 250 kPa. Z will have water potential of negative 750 kPa. If you remember concept of water potential, water molecules will move from a region of high water potential to region of low water potential across semi-permeable membrane without using energy. So daripada yang paling tidak negatif pergi ke tempat yang paling negatif. So between X and Y, water will move from Y to X. Between X and Z, water will move from X to Z. between Z and Y, water will move from Y to Z. Jadi berdasarkan anak panah yang kita sudah lukis di sini, saya tengok option A, B, C, D. The only option is 3C. X to Z, Y to X, Y to Z. Kalau masih confused, coba lukis lagi, coba lukis sel-sel itu di atas kertas apa-apa and try to look at the movement of water molecules. Any problems? Okay, so I don't think we have any problems, so I'm gonna continue. Right, so I'm continuing with question number 4. Ada yang keluar masuk. I hope you're able to follow the lesson still. Okay, so question number 4. If two sucrose solutions of different concentrations are separated by a partially permeable membrane, which of the following will occur as a result of osmosis? So ini dia lebih kurang tengok apa yang kamu faham pasal osmosis lah. Okay, let's look at option A. The solute will diffuse from the more concentrated to the less concentrated solution. Before I talk about whether this is correct or not, I just want to remind you that solute is the sugar or the salt, whatever yang dilarutkan. Solvent is the water molecule or benda pelarut. Okay, so how I like to remember is solvent, V macam water. So I try to remember that. Solute will diffuse from the more concentrated to the less concentrated solution. Maybe. Tapi kalau semi-permeable membrane, biasanya solute yang kita gunakan itu tidak dapat merentas. If it is salt, it can cross. But this one is sucrose solution, sucrose gula. So sucrose molecules are bigger. They usually cannot pass through partially permeable membrane. So A is incorrect. For B, solute and solvent will diffuse until equilibrium is reached. Nope. Solvent can diffuse, solute cannot diffuse. Between C and D, volume of the less concentrated solution will increase. Or D, volume of the less concentrated solution will decrease. So for this one, you should know that the less concentrated solution, the solution that contains less solute, will have higher water potential. Less concentrated solution will have higher water potential. Sorry, that's not a symbol for water potential. This is water potential. So if it is the region with higher water potential, dia akan kekurangan molekul air. So the answer for 4 is D. Any problems with this one? Okay, then I am continuing with the lesson. Okay, continuing with question number 5. Which of the following organelles in animals are surrounded by double membrane? Answer is A, nucleus and mitochondria. Golgi apparatus is only one membrane, single membrane. Organel-organel dalam sel kita boleh dibahagikan kepada organel yang ada satu lapisan membrane dan organel yang ada dua lapisan membrane. This is important if you want to learn more about cell biology in university, or if you're learning things about like evolution and where life came from and why bacteria have this kind of organelle but eukaryote have this kind of organelle. So yeah, yang ada dua lapisan membrane, nucleus, mitochondria. Okay, question number 6. Why is the formation of chiasmata an important feature in meiosis? I just want to remind you what is chiasmata. So when you have your homologous chromosomes pair up, they do the synapsis during meiosis. Okay, yang dirang berpegang tangan. The area where the two non-sister chromatids meet and exchange genetic information, this is your chiasmata. Chiasmata. Okay, just to remind you, that is chiasmata. So why is chiasmata important in meiosis? Question number 6, answer is C. It will provide opportunities for new genotypes to arise. A salah, B salah, D salah. Any questions? Okay, no questions, so I'm going to continue. Question number 7. In which of the following events is observed during mitosis? Question number 7, answer is B. Options 1 and 3. 1, movement of centrioles to opposite poles is correct. 3, appearance of chromosomes with 2 chromatids joined together at the centromere, correct. Exchange of genetic materials between non-sister chromatids, not correct, because this one is mitosis. Any problems with this? Any part you don't understand?
Tidak, Miss.
Okay, that's good. Then I'm going to continue. Right, question number 8. There are 40 units of DNA in the cell during stage G2. How many units of DNA are in the cell during G1? So this is, oops, sorry, wrong marker. So if you can identify very clearly, this is from chapter 3, cell division. Cell cycle is divided into 4. You have your G1, you have your S phase, you have your G2, and then you have your M phase. So kalau semasa G2 ada 40, that means semasa S phase dia sudah berganda. Sebelum dia berganda, it is going to be half the number, which is 20. So answer for 8 is B. Should be very simple. I'm moving on to question number 9. Question number 9, we already go into chapter 4. The genes for shape of seed and texture of seed coat are located on non-homologous chromosomes. Dia bagi tahu non-homologous chromosomes, so ini adalah not linked gene. Which of the following crosses will produce F1 offspring with heterozygous genotype? Big R, small r, big S, small s. So daripada 3 ini, adakah dia dapat menghasilkan genotype ini? Kalau dia dapat, walaupun F1 generation dia bukan ikut nisbah begini atau dia bukan semua heterozygous, ada yang homozygous, ada yang tidak, it still counts. So all 3 of these can still produce this genotype. So number 9, answer is D. Okay, if we have no problems, I'm moving on to question number 10. Question number 10. Of a normal couple, half the sons are hemophilic while half of the daughters are carriers. The gene is located in, before I want to answer this, I want to try drawing the family tree. So you have the father and the mother, kawin. They look normal, so they're not hemophiliac. And they have several children. Half of the sons are hemophilic. Okay, so square is for boys. Half of the boys will be healthy, half of the boys will be hemophilic. So I color that full color. They also have daughters. Half of the daughters are carriers. So one daughter healthy, another daughter is carrier. So now we have to find out where did that hemophilic gene come from. So X will be normal. X small h will be hemophilia. Y is just Y chromosome lah. So let's look at the dad. The dad will be sama ada XH ataupun XY. Oh wait, let's look at the children first. The children, the healthy son is gonna be XY. Hemophiliac son is gonna be XHY. Healthy daughter is gonna be XX. Carrier daughter is XXH. So this is the possible genotype of the parents. And the answer for question number 10 is going to be C. The gene for hemophilia came from one X chromosome of the mother. Any questions?
Gender miss.
Yeah. Okay, very good. Then we'll just continue. All right, so question number 11. An extra finger in humans is rare, but is due to a dominant allele where one parent is normal and the other parent has extra finger but is heterozygous for the trait. What is the probability that the first child will be normal? So if you have the first parent with normal hand, 1, 2, 3, 4, 5. Okay, but the other parent has extra finger. 1, 2, 3, 4, 5. 5. So this one is gonna be small f for the parent with normal fingers, normal number of fingers, and big F, small f for the parent with extra finger. What is the probability that the first child will be normal? Normal means they are small f, small f. So if we let them cross, the ratio of their children is gonna be half of them will get extra finger and half of them will get normal fingers. So answer for question 11 is C. 50% of their children will have normal fingers. Okay, I'm moving on to the next question. Question number 12. Which blood type would be possible for children of a type AB mother and a type A father? So this is the type AB mother and type A father. So type A, I hope you remember apa dia punya possible genotype. Father could be this or this. Mother is most definitely IA, IB. So question number 12. Only possible answer is C. This one should be simple, huh? But if you're still confused, feel free to ask or try to make your own genetic diagram. Okay, I'm moving on to question 13. Okay, so question 13 and 14 is for chapter 5, which is my favorite chapter. I'm not sure if I told you already, but it's my favorite chapter. Very easy to get marks as long as you remember where to start. In a population that is Hardy-1 equilibrium, 16% of the individuals show the recessive trait. What is the frequency of the dominant allele in the population? Calculate up to 3 decimal places. So what we're going to do first is we're going to take out the information. Okay, so here 16% of the individuals show recessive trait. So that means 16% is our Q square. Question also asks, what is the frequency of the dominant allele in the population? Frequency of dominant allele is P. So all you have to do is start with Q square. Q square is 16%. You can immediately make that 0.16. So to find Q, you use square root. Means Q is going to be 0.4. And then guna formula P plus Q equals 1. P equals 1 minus Q. P equals to 0.6. So the answer for 13 is very simple. It's just C. 0.6. Okay, I'm just checking something. Okay. Can you hear me talk? Okay, you can hear me talk. was double checking on my phone. Okay, question number 14. 12% of the babies are born with sickle cell anemia. What is the frequency of the carrier genotype in the population? Okay, so for this one, 12% is the Q square. Frequency of the carrier genotype in the population is 2PQ. So we just start with Q square again. Q square is 0.12. To get Q, you need to find the square root of 0.12. So you take your calculator. And you tekan, and you should get 0.346 for Q. So that means P is gonna be 1 minus 0.346. .654. And then what you need to do last is 2PQ is equal to 2 times 0.654. And you're gonna get 0.452 ataupun 453. Okay, so I got 4525. 526. Tapi yang paling dekat punya inilah. So 14 would be C. Okay, jalan kerja sudah ada di sana. Shouldn't be any problems. So I'm just going to continue with question number 15. So, question number 15. Masih hidup lagi kan kamu? Okay. So the way this text is laid out, I'm gonna separate it so it looks a bit easier to read. Okay. So the normal gene sequence in a chromosomal sequence is A, B, C, D, E, F, G, H. And then the question gives you 3 scenarios. If A, B, C, D, E, F, G, H is changed to A, B, C, G, H, this one is?
Deletion.
Very good. It's deletion. If it is changed to A, B, C, D, E, F, G, H, what has occurred?
Duplication.
Yes, very good. So this one is duplication. And if it is changed to A, B, C, F, E, D, G, H, this one is?
Inversion.
Yes, very good, Sheila. Thank you very much. So this one should be very simple. Lepas kamu isi tempat kosong, dapat sudah dia punya jawaban. Deletion, duplication, inversion. Number 15 is A. All right, moving on to question 16. If the DNA codon ATG, 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 and cytosine base is inserted at the beginning, which of the following will be the result? So for question number 16, it should be C. So kamu nampak tiga-tiga ini adalah frame shift mutation. Cuma cara dia susun codon itu tidak berapa betul. Remember that your genetic code will be read in groups of 3. So ini dia punya pembagian 3 tidak betul. These langsung tidak berkenaan. Okay, so we're continuing with... Oh no. My word is frozen. But you can still see the question number 17. A segment of DNA has base sequence AAG, GAG, GAC, CAA, CCA. Which of the following sequences represents a frame shift mutation? So for question 17, kamu tengok frame shift mutation, it is gonna affect more than one codon. Kalau dia bagi efek kepada satu codon saja, means it's just missense mutation. It has to affect more than one codon after the point of mutation for it to be considered a frame shift mutation. So the answer for 17 will be A. Okay, my laptop hangs kejap, so I'm just going to close it for a little bit. I'm back and I am continuing with my share. Sorry, sa punya laptop kadang-kadang hang. So I have to put it to sleep and then reopen it. Okay, so yeah, like I said just now, the answer for 17 should be A. Any questions, any problems? Okay, should be good. And then I continue with question 18. Question 18 kita masih lagi di chapter 7, but this one is 7.3. So this is already about chromosomal mutation. Which of the following are examples of diseases that can be caused by non-disjunction? If, in case you forgot, non-disjunction is when the chromosomes or the sister chromatids are not able to separate properly during anaphase, ataupun anaphase 1, ataupun anaphase 2. So between the three of these options, the only answer is 1 and 2. Turner syndrome and Klinefelter syndrome. Hemophilia is not the result of non-disjunction. Hemophilia is the result of gene mutation. So 18 is A. Should be no problem. I'm continuing with question number 19. Which of the following would be the most suitable vector to carry a target gene worth 40,000 bases? So ini dia macam saya tidak tekankan lah masa kita belajar, tapi memandangkan kita buat ni online kan memang hari tu ada apa kan kamu buka lecture note. So if we look at the lecture notes, let me try to copy the part from the lecture note into here. Okay, here. I want to copy this. Can I put this in here? Yes, I can put this in here. So if you look at this thingy from our lecture note, yang dapat tampung 40,000 bases adalah cosmid. So number 19 would be C. Would be B, cosmid. Look at this, ini sini ada tulis K kan, so this is kilobase, so that is actually 35,000 to 50,000 bases. 8,000 to 20,000 bases. So the most suitable one is cosmid lah. Boleh juga kalau kamu mau guna YAC, tapi bila benda itu terlampau besar, macam susah kamu mau kawal dalam eksperimen kamu. So the most suitable one is the one yang ngam-ngam in the range. Okay. Was there a question just now? Any problem, Ebiliska?
Miss.
Okay, kalau tidak, then we just continue. All right, so continuing with question number 20. Which of the following requires the production of cDNA? cDNA is what you make when your gene of interest is from eukaryote. You want to put it into prokaryote host. 20 is C. Question 21. The presence of white and blue colonies in the blue-white screening indicates A. Transformation process was successful. B. Insertion process happens at 100% success rate. C. White colonies originates from host cell transformed with plasmid carrying intact, like Z. D. Blue colonies originate from host cell transformed with plasmid carrying disrupted, like Z. Kalau kamu tidak belajar pun, kamu baca soalan ini betul-betul, pun kamu dapat teka mana satu jawapan yang betul. So for question 21, option is gonna be, the most correct option is gonna be A. If you have colonies during your blue-white screening, that means the transformation process was successful. It is not 100% successful, sebab itu ada yang biru, ada yang putih. Dan explanation kenapa dia biru, kenapa dia putih, terbalik untuk C dan D. So sepatutnya yang white colony adalah disrupted like Z. Blue colony is intact like Z. Okay for this question? Okay, then I'm gonna continue with the next question. This is a bigger diagram. A gene cloning experiment was carried out by using the plasmid PUC18 as the cloning vector, which is still the normal plasmid that we always learn about. The screening process will utilize which component? So screening process adalah yang blue-white screening itu. Okay, dia akan tengok bahagian mana pada plasmid. So the answer for this is, when we're doing the blue-white screening, we're looking at whether or not it received a plasmid. If it received any plasmid at all, it will have ampicillin resistant and it can survive in the medium containing antibiotic. And if it has plasmid, then we look at what color is that colony of bacteria. And what color it is will tell us if the gene of interest can be put into the leg Z. So the answer for question 22 is D. Blue-white, the screening process will utilize the ampicillin resistance gene and the leg Z gene in the plasmid. Any questions with this one? Okay, then if no question, I'm going to move on to question number 23. This one might be a little bit confusing. A host cell has successfully taken up a recombinant PUC18 plasmid, but the plasmid bears mutated origin of replication. So dia punya origin of replication is mutated. Which of the following will happen to that host cell following amplification process on a media containing ampicillin and X-gal? Remember, amplification process is when we let the bacteria divide and grow so that it can produce more of that gene that we put in and produce more of the gene product. So between options 1, 2, and 3, you need to look at the choices, sama ada 1 dan 2, 1, 3, 2, 3, ataupun semuanya. Who can tell me what is the effect on the host cell or on the plasmid when the plasmid's origin of replication is mutated? We focus on this sentence now. Plasmid dia ada mengandungi gene of interest, ada mengandungi ampicillin resistant gene, tapi dia punya origin of replication rosak, mutated. So what's going to happen? Anyone? Shifa terkejut sampai dia left. No, just kidding. That is internet problem. Anybody? Anybody? Cuba teka kah? What happens? Nobody at all? Is this why you guys didn't get full marks? Can Miss repeat the question? Okay, the question is, what happens to the plasmid or the host cell after the origin of replication is mutated? Origin of replication dia rosak. So apa kesan dia kepada plasmid dan kepada host cell? Okay. Akil cakap dalam chat cannot replicate. Which one cannot replicate? What cannot replicate? Is it the plasmid? Is it the host cell? Or just the gene of interest? Elot teka, the correct answer is 1 and 2. Oops. Oh, don't tell me jam again. Okay, belum. Saya ingatkan hang lagi. Okay, so good guess. It survives, but it cannot replicate. Elo cakap it survives, but it cannot replicate. So let us try to look at this situation. Man, my pen is not doing good today. You have your host cell. This is your E. coli. And this is your plasmid yang ada gene of interest. Tapi saya malas kasih tukar-tukar the ink. When it is ready, this is gonna divide by mitosis and produce 2 E. coli cells. So the host cell is able to replicate, but the plasmid is not able to replicate. Plasmid dia sahaja. So only one of the daughter cells of the E. coli is able to receive the plasmid. Since the other one does not have plasmid, that means when it reaches the medium, bila dia keluar, because the medium still contains antibiotic, if it doesn't have plasmid, it doesn't have the antibiotic-epicillin resistant gene, it's gonna die. So the other one will survive, but when it comes the time for it to replicate, the same thing will occur. You're gonna have 2 cells. But only one of the cells will be able to receive the plasmid. So this one is gonna die juga. So yeah, so it's not gonna be able to form a colony. It survives in the medium, but only one of it. It will not be able to digest X-gal. Will it be able to digest X-gal? It should be able to, but cannot form colony. I'll just double check this one. No, wait. It would not be able to digest X-gal. It would not be able to digest X-gal. Number 3 should be correct. Because this is successful recombinant plasmid. Successful recombinant plasmid means that the leg Z is not function. Yes. Yeah. I'm confused. All right. So yeah. So question 23, 1, 2, and 3. So it should be D. Thank you, Elot. Thank you, Akil, for answering in the message. Any more problems? Any questions regarding question number 23? Okay, should be all right. So I'm going to continue with question number 24. During production of cDNA, why does the cDNA mRNA hybrid must first be treated with RNAs before the addition of DNA polymerase III? So to help you like kind of make sense of the question, mungkin ada yang English dia tidak berapa campin, so dia terconfused di sini. So when you are making the cDNA, you start with the mRNA kan? You started with mRNA. And then later you're gonna have mRNA and cDNA. Okay, purple is mRNA. Green is cDNA first strand. And then you're gonna use enzyme RNase to digest the mRNA so that you only have the cDNA. cDNA first strand. And then after that, you use DNA polymerase to get 2 strands of cDNA. Sini first strand, ini second strand. So at this step, you use reverse transcriptase. Okay, so itu ringkasan apa yang berlaku semasa penghasilan cDNA. So the answer for question number 24 is 1, 2, and 3 will be correct. The reason why we use RNase is to free the mRNA so that it can be reused again by reverse transcriptase. Wait, oh, sorry, no. 24 is C, 24 is C. Silap siya. 24 is C. Sebab lepas number 1 is not correct because after you use the RNase, itu mRNA dia kena break down into fragments. So it cannot be reused. It is to degrade the mRNA and to free the cDNA so that cDNA can be used as template strand to produce second strand of cDNA. Any problem with question number 24? Okay, you should be all right. Then I'm just going to continue with question number 25. The growth of pollen tube towards the ovary is influenced by... Anybody have the answer to this? This is something I mentioned before in the class. 25, anybody have the answer? D, yes, correct. So it is influenced by the chemical signals released by synergid cells. Sebab masa itu kita sudah cakap pasal apa fungsi synergid cell untuk membimbing pertumbuhan pollen tube. Okay, so let's continue. Question number 26. A woman is said to conserve the number of her primary oocytes when? Conserve number of primary oocytes means dia tidak gunakan, dia tidak membazil dia punya primary oocyte, yaitu ovum yang belum matang itu. So the only time when you pause development of your primary oocyte and you don't waste it is when you are pregnant, if you're a woman. If you're a guy, you don't have oocytes. Okay, so 26 answer is B. If you abstain from sexual intercourse, you don't have the sex, you don't get the pregnant. Every month, the primary oocytes will still mature one by one. When you hit menopause, that means you already finished all your primary oocytes. When you are having your period, that means you are getting rid of your secondary oocyte. Okay, so only correct answer is 26B. She's pregnant. And it's going to continue developing the rest of the primary oocytes after pregnancy is over. Okay, so question number 27, still on chapter 9. A developmental error during pregnancy has caused a child to be born without body hair and problematic skin. Predict the germ layer that is involved. So body hair, problematic skin. You try to remember, is this relating to the ectoderm? endoderm, or the mesoderm. Cara yang simple saya kasih ingat kamu hari itu, saya ada lukis itu orang kan. I draw a person for you. This layer and including the brain, the saraf-saraf, your nerves, and your akar tunjang, your spinal cord, is part of one layer of tissue. Your muscle, your other internal organs, that's gonna be part of another layer. And another layer is just the lining of your gastrointestinal tract, lining of your respiratory tract. So from your mouth, from the inside of your mouth into your lungs, and also from the inside of your mouth all the way to the inside of your butthole. So those are the three germ layers. So 27, it should be A, ectoderm. Because this is relating to the body hair and skin. So itu ada lapisan yang biru. Any questions regarding these? Any problems? Mendekat habis sudah. Okay, then I'm gonna continue with question number 28. Which of the following refers to the hormones released by the anterior pituitary gland? Can anybody tell me what are the hormones released by anterior pituitary gland? Ada dua jenis, tolong namakan. Tolong namakan dua hormon yang dihasilkan oleh anterior pituitary gland. You can give me the short form juga. FSH and LH, correct. So we need to look for which curve corresponds with FSH dan LH. Number 1, what do you think is number 1? Is this FSH, LH, progesterone, estrogen? Curve number 1. Yang lebih tinggi sepatutnya FSH kah LH? Oke, yang lebih tinggi sepatutnya LH. Yang lebih rendah, tapi dia macam naik lebih kurang pada masa yang sama, which is number 3, is gonna be FSH. Okay, a good try. Okay, good try. Intan, Nadi, Alot answered correctly in the chat. So other than FSH and LH, you also have estrogen, which is curve number 2. And the last curve, the dotted line one, is progesterone. So this one is progesterone. Okay, so hormones released by anterior pituitary gland is 1 and 3. Kalau kamu bingung, kamu tidak dapat kenal pasti mana satu LH, mana satu FSH, mana satu estrogen, mana satu progesterone, kamu tengok saja, anterior pituitary gland hasilkan dua hormon ini pada masa yang sama. Jadi dia akan meningkat dan menurun pada masa yang lebih kurang sama. So the two curves here that are going up or going down at the same time or around the same time is 1 and 3. So kalau main teka pun, kamu boleh teka 1 dan 3. Okay, so question number 29. The diagram shows the cross-section of a seed. Which of the following will have altered chromosomal number if fertilization takes place between abnormal first sperm nucleus with a normal egg cell? So dia tanya, kalau ada abnormal first sperm nucleus and normal egg cell, part yang mutated, yang pelik itu, dia akan membesar jadi bahagian mana dalam benih ini. So you know that the abnormal first sperm nucleus with normal egg cell will become the zygote. The zygote will eventually become the radicle and the plumule. Radicle and plumule, or in other words, it becomes the young baby parts of the new plant. Okay, this is the baby of the new plant. So it's actually going to be this. Yang macam daun ini adalah dia punya plumule. Yang macam akar ini adalah dia punya radicle. So it's gonna, the part that will have altered chromosome number is 2 and 3. Question 29 is C. Any problems with this question? Bagian satu ini apa? Apa nama dia? What's that big thing in the middle there? What is this called? Anybody know what is the thing in 1 called? Yes, that is the endosperm that becomes the nutritional tissue. Ini adalah dia punya endosperm yang akan bekalkan radicle dengan plumule dengan nutrients. So it's going to supply nutrients to the young plant until it's able to photosynthesize on its own. So yes, correct. That's the endosperm. All right, very last question. So I mentioned this earlier in the week when I talked about Chapter 10, growth. At which point of growth would be most critical if you're the owner of a seafood restaurant popular for soft-shelled crabs? So, soft-shelled crab, if you ever eat it or not, is not a different species. Dia bukan baka yang berbeza untuk ketam. Dia adalah ketam yang baru sahaja molting, baru sahaja ecdysis, means dia baru buka kerang dia yang lama. Dia punya isi masih lagi lembut sebab dia punya kerang baru belum siap develop. So, if we are looking at the growth pattern of the crab, I mean, you don't have to draw the growth pattern here, but I want to show it to you anyway. Then you're gonna have the steps like this. This is gonna be the size over time. Part yang saya highlight ini adalah ecdysis ataupun molting. This is ecdysis, also called as molting. This is called an instar. So the time that it spends not molting is called instar. Oops. Instar. So this is instar 1, instar 2, instar 3. Bila dia sudah paling matang sudah, when it reaches the adult stage, it can be called adult, it can be called the imago stage. Crabs, do we call them nymphs? We don't really call them nymphs. At some stage adalah. But most importantly for this question, what you need to know is soft-shell crabs is if you catch the crab and you cook it during its ecdysis stage. So question number 30, answer should be C. Any problems with this? Okay, that should be it for question 30. Do you have any problems at all with any of the questions? Anything else you want to ask before I finish the discussion? Discussion ko nonsense yang cakap. Excuse me, I think I burped. Any questions? Any soalan? Tiada. Okay. You're all okay. Alright, if you're all okay, then should be no problem. Saya yakin ada dari kamu, saya harap kebanyakan dari kamu lah yang sudah start buat soalan soalan praktis, soalan daripada college lain kah. So yeah, go ahead and do that. Especially since it's objective questions, it should be fairly easy for you to do your revision. Okay, yeah, so I'm gonna bring you to the announcement part. So good question, Elot tanya dalam chat. PSPM objektif nanti di paper ataupun di Google Form. Turns out it is on paper. Hari itu saya cakap Google Form kan, tapi rupanya saya silap. PSPM kamu bagi yang berada di college ataupun yang akan datang semester depan, kamu akan menjawab PSPM secara bersemuka dan jawab atas kertas. Hari itu saya yang salah paham. So once again, 9 to 16, your biology paper is on the 14th of December. So that is the Tuesday. It will be at 9 a.m. to 10 a.m. The full schedule for your PSPM is already in the Telegram group. I forwarded to the chat version of the Telegram group. So nanti kalau kamu mau forward antara kamu, feel free. 30 objective questions, a lot like today's lesson. You need to come in with just your writing tools, your IC or your CAD metrics, wear mask or whatever. Datang begitu sajalah. Bawa calculator. Kalau tidak silap semua subjek, jawab secara fizikal, yes. Sepatutnya ada taklimat exam untuk pelajar juga. It should be sometime next week. Week-ish. It will be online. Tunggu memo daripada college bila itu taklimat exam. Taklimat exam daripada pegawai pengurusan peperiksaan Encik Landan. Bagi yang berada di college sekarang, lepas exam, so starting from the 17th of December, kamu cuti. I'm not sure when is your muet, but you have to come back here for your muet. I think you should be able to go home during the holiday, but we have to see how the situation is all over the country in Labuan. And also, kalau ada memo daripada bahagian kementerian lah. Hopefully, everybody can go back to their kampung, whatever, and enjoy your holiday. But don't be surprised if anything changed. Palis-palis. Harap boleh balik lah. And after holiday, come back from wet. Those of you who are at home right now, you will come back here from wet. And after your wet, then you will take your PSPM 1, physical, in-person, duduk di Dewan or Dewan Kuliah or whatever. I don't know why I hold my breath when I talk to you, so now I'm out of breath. Any questions regarding the PSPM punya stuff? Stop presenting this one. Okay, a few more announcements for me to make. Miss, sem depan kelas face to face sudah lah tu kan? Not completely face-to-face, but some classes will hopefully be face-to-face, yeah. Okay, so what I wanted to tell you is do your revision. There's some revision structure question. There is a practice multiple choice questions for you to answer. This one is a set of multiple choice questions. This one is 40 questions lah. I think ini termasuk dengan chapter 6. Yeah, this one is termasuk dengan chapter 6. Itulah dia ada 40 soalan. But you can still try to answer. And if you go to this website later, we have this strange picture thingy. So if you go to this PDF, we have collected questions for you for each chapter. So you can go to the PDF, press, and it will direct you to the PDF for that chapter. So contoh ini adalah koleksi soalan untuk chapter 1 dengan jawapan di hujung. So kamu boleh cuba buat sendiri, tanda sendiri. Chapter 2 pun ada, chapter 3 we have 11 questions, chapter 4 we have 24 questions, chapter 5 we have 8 questions, chapter 7 38, chapter 8 32, chapter 9 24, chapter 10 8 questions. So kamu boleh practice banyak-banyak kali lah. Hopefully that's helpful for you. And okay, also here I also have, this is the jadwal PSPM yang ada saya forward dalam group. So boleh nampak sini, your class is gonna start from, no, your exam is gonna start from 9 December with maths, 13 December chemistry, 14 December bio, 15 December computer science. Physics tu kamu bebas sudah. So you can use that extra day for packing or whatever. Any other questions? Any more problems? Okay, what else do I need to ask you? Miss, modul 3 exam di mana? Kamu akan dapat tahu mana tempat ambil exam kamu nanti. So later you will have, sometime next week you will have taklimat exam online. So taklimat exam tu diorang akan bagi tahu, okay, apakah peraturan dia, apa bahan terlarang, apa yang boleh dibawa, apakah etika pemakaian kamu. But basically pakai formal untuk pergi kelas lah. And later you will also get your exam slip. So slip exam akan ada maklumat kamu di mana dia akan tulis kamu punya nama, number matrix, IC, modul, dan tempat duduk. So based on your exam slip, you will have your number, tempat duduk, dan pusat exam di mana. So wait for that information. The pusat exam is either in bilik tutorial, library, ataupun Dewan Mutiara, which is the big Dewan near the ATM. Any more questions? Oh, okay. I also want to know if you are, how many of you are still at home and how many of you are here. I was trying to do a poll on Telegram, but when I do it in the channel, it's only anonymous. And when I do it in the chat group, I couldn't see other people responding. So maybe I'll try that again later. Nanti bila saya send poll di Telegram tu, mohon kerjasama kamu untuk jawab lah. For those of you who are revising, maybe you want to do extra notes kah, ataupun kamu mau jawab past year, kamu mau saya tanda. Yes, feel free to private message me. You know me, it's very easy for things to slip from my mind. So if you want me to mark anything, PM me and basically peer pressure me into doing the work. If you already asked me to mark something before and I haven't done it, please remind me. I'm old. And then if you're doing any kind of revision material, you get stuck, you want to ask me question, tanya saja. Walaupun yang dalam college sudah habis PSPM, kamu berada di rumah, kamu masih mau tanya saya soalan, message saja. Even if I reply late, I'll message you. Walaupun waktu cuti pun, says I finish whatever I'm doing, I'm gonna reply your message. So don't worry about that. I get paid to answer your questions. So don't feel guilty. Message saya 3 pagi pun, don't worry. When I wake up, I'll message you back. There is one more thing I need to send to you today, which is the quiz number 2 for 9.1. Hari itu 9.1 kan saya cakap saya mau test sama ada kalau saya bagi kamu flashcard dengan guided drawing, adakah itu dapat membantu kamu untuk lebih memahami konten dalam 9.1? So I'm gonna send you the quiz 2 later to see if you improve a bit in your understanding. Any more questions? Any more stuff? If you have any problems while you're in college and it's something that I can do, like don't ask me to add Wi-Fi in your dorm. I cannot do that. But if you need me to buy like chewing gum and stuff, ataupun spek rosak ka, and you need me to send it to the shop, that one I can do. Are we good? Are we okay? No question, no more problem. Sure. Okay. So if there are no more problems, those of you...
No, miss.
Okay, thank you, Intan. So if it's no more problems, those of you who are willing to open your camera, can you open your camera so I can take a screenshot and I can also post things on social media? Hi, Uzail. And like, it's not just my friends who can post things on their Instagram. I want to do it too.
Hi, Miss.
Hi. Hi, Kila. Hi, Sheila. Hi, Akil. But some of you, your internet may not be good enough, and that's okay. Kalau tidak dapat pun tidak apa. Hey. Yes, I can make Facebook. I can make Insta posts. Miss, count.
Tidak,
I need to
Miss.
count. 1, 2, 3 kah? 3, 2, 1. Okay, 1, 2, 3. Okay, let me take a snapshot. Yay! I hope I get to see you guys face to face next semester. Okay, thank you. Saya sudah gambar. Okay, so
Thank you,
that's
Miss.
thank you. So that's pretty much it. Lepas daripada ni, usaha kamu sajalah untuk study untuk PSPM. I'm sorry if I haven't been doing much for you this semester. I'll try to do better next sem. But yeah, let's just do our best from here. Oh, almost forgot. One more thing. Nanti ada exit ticket, which is another form you need to answer to tell the college if I teach you good enough or not. Kalau boleh bagi score yang tinggi sikit daripada entrance ticket hari itulah. I'll post that in the GC later. So yeah, so daripada sini, dari usaha kamu saja, I'll try to help where I can. But yeah, whatever happens, happens. I wish you all the best. Make sure you manage your time and your stress properly. Don't forget to eat, even though cafe food is expensive. At least makan lah, jangan sampai gastric naik. If you need help in any way, feel free to ask me for help, ask other lecturers for help, ask the counselor, ask friends. us family, etc. Thank you very much for the pleasant one semester teaching you. And yeah, wishing you all the best. And I'll see you guys again soon. Bye.
Thank you, Miss.
Thank you.
Goodbye,
Miss.
Miss.
Thank you. Bye.
Thank you,
Thank you,
Miss.
Miss.
Thank you, Miss.
Thank you, Miss.
you.
Thank